Today we will study about the microscopy. I am Dr. Humaira from J.A.T. Art Science and Commerce College for Women Malegaon Nashik. My topic is microscopy, in which we will study about simple and compound microscope. So microscopy is the technical field of using microscopes to view objects and areas of objects that cannot be seen with the naked eye. Thus, for visualization of microorganisms, an optical instrument called microscope is used. Microscope is a basic tool of microbiology. It magnifies the size of image of an object. So, based off use microscopic is microscopy has many types of microscopes, among which is based on the source of illumination. So light microscope and electron microscope is used usually. In the light microscope, usually two microscopes is used. One is simple, another one is compound. The simple microscope is a scientific instrument that makes things that are too small into big so that they can be examined correctly. A simple microscope is a microscope that uses only one lens for magnification. It is the original design of the light microscope. The use of single convex lens for viewing is still found in simple magnification devices such as magnifying glass and loupe. So, the simple microscope consists of these parts, that means light source, diaphragm, stage, objective, resolving nose scope, body tube, and ocular lens. The working of the simple microscope takes place like a refracting telescope but with minor differences. A telescope must gather a large amount of light from a dim, distant object, and so it needs a large objective lens to gather as much as light as possible and bring it to a bright focus. Since the objective lens is large, it brings the image of the object to a focus at some distance away, which is why telescopes are much longer than microscope. In contrast to telescope, a microscope must gather light from a small area of thin, well illuminated specimen that is close by. So, a microscope does not need a large objective lens, and so it is small and spherical. It has much shorter focal length on either side. It brings the image of the object into focus at a shorter distance within the microscope's tube. The image is then magnified by the second lens known as ocular or eyepiece as it is brought to the eye. A microscope has a light source and condenser. The condenser is a lens system that focuses the light from source on a small bright spot of the specimen. Microscopes have interchangeable objective lenses and fixed eyepieces. So this is about the simple microscope, in which the above the base light source is present, which collect the light from the sun rays or from the local light or from the bright background, and by the help of diaphragm, the adjustment of the light takes place. Objectives are helpful for seeing the objects or for the formation of the image, and by the help of the eyepiece we can see objects in large size. So in such a way simple microscope work simply. The next type of microscope we will study today is nothing but the compound microscope. In the compound microscope, the light is immersed and transmitted and focused by minor in condenser. It passes light as a cone of light so as to illuminate the object or specimen. The refracted light is collected by an objective where primary image of the object is formed. This is real, inverted, enlarged image of the object. It is formed in the body tube. This image is taken as an object for an eyepiece. The eyepiece further magnifies this primary image into virtual erect enlarged image. This is the final image that lies above the stage. As the primary image falls before F of the eyepiece forms virtual erect image. The most commonly used microscope for general purpose is the standard compound microscope. It magnifies the size of the object by a complex system of lens arrangement. It has a series of two lenses, objective lens close to the object to be the observed, and ocular lens or eyepiece through which the image is viewed by eye. Light from a light source, that means mirror or electric lamp, passes through a thin transparent object. The objective lens produces a magnified real image of the object. This image is again magnified by the ocular lens or eyepiece to obtain a magnified virtual image, that means final image, which can be seen by eye through the eyepiece. As light passes directly from the source to the eye through the two lenses, the field of vision is brightly illuminated. That is why it is a bright field microscope. The object is mounted for the seeing image. The working of the microscope is in such a way the object is mounted on the stand below the microscope tube. The objective lens forms a real inverted and magnified image of the object. The image acts as an object for the eyepiece. The position of the eyepiece is so adjusted that the image lies within the focus of the eyepiece. The eyepiece acts like a magnifying glass and forms a virtual, erect, and magnifying image of the objects. Image formation in a compound microscope takes place as the object is placed just outside the principal focus of the objective lens. The principal focus of the objective lens on which a inverted magnifying image is formed. The magnified image acts as an object for the eye lens, and the final image is virtual and magnified still further. It is inverted compared with the object and may be 1000 times larger than the objects. In such a way, the compound microscope work and make the objects larger as its original size. So in such a way the microscopic work is completed.